Thanks, John. Good morning, everyone. I've just gotten off an 11-hour flight, so I'll try to stay coherent here. As you can see, my name is Jennifer Gardner. I'm here from the Gates Foundation. It's not actually true. When we set this meeting up, we wanted to have, the Gates Foundation is running a TB biomarker program, and we thought it would be nice to have the Gates Foundation basically describe that program to you. But unfortunately, Jennifer and her colleagues weren't available. So Jennifer's kindly given me her slide set, so I'm actually sort of presenting a Gates Foundation presentation, not really a fine one. The Gates Foundation has set out strategic goals for TB control and management through research efforts. As you can see, they have sort of aspirational goals, eliminate TB, routines goals that are not sufficiently ambitious, perhaps just to reduce TB mortality, and really their principal goal, which is they think achievable, but focused and perhaps will have the most immediate. Told you I just got off the plane. Impact would be to accelerate the reduction in incidence by other means, including other means through early detection and stopping transmission. This is where their biomarker activities fit in. As you know, they're working across a number of both implementation areas as well as on development of new tools, including vaccines, drugs, and diagnostics. The diagnostics goals they have for the 2011 to 2016 period is to identify one new TB biomarker, that's one validated, functioning, useful TB biomarker, and two new molecular diagnostics that would be endorsed by WHO STAG if they perform well enough. So those are the goals for the coming now 3 years. The diagnostics work from the Gates Foundation is primarily focused on R&D activities and with a small component of implementation. The diagnostics timeline for their activities are worth talking through. For molecular diagnostics, the expert activity, their involvement, besides funding us to work with Cepheid and the Elan group to develop the assay, their involvement included helping reduce the price down to the current sub-$10 price, as well as country support for uptake. In the same time now, more recently, they are looking at funding the development of alternative or competitive approaches. You may have seen, many of you, they've given a large grant to Lear to develop something on their QPOC platform. They are intending to develop this ideally by the end of 2014. They are going to be looking for a second candidate, an alternative additional molecular platform. They have also put out an RFP in China, and as that's now been completed, and they've had a number of interesting proposals. FIND is going to be working with one of those groups that looks like they're going to be funded to develop a point-of-care molecular platform. The TB biomarker work, which is what we're talking about today, primarily involves an RFP, which will describe the process, a number of phase 1 grants, and then some phase 2 grants of those that seem to be most promising. I won't tell you what's wrong with the existing solutions. I think this crowd doesn't need that slide. So the program goals for the biomarker work at the Gates Foundation include to identify and validate biomarkers that will enable the development of simple point-of-care assays like the HIV tests are currently. Biomarker investigation would be focused on blood, urine, and breath samples, that really trying to avoid sputum as the matrix. Besides just a diagnostic test, a triage test, one that allowed you to detect essentially all patients, even if not with high specificity, so you could refer those patients for confirmatory testing, would be also inside of scope here. Drug resistance is not explicitly included within the scope of the program. So this program is a two-phase program with about $12 million set aside for it. There were, when Gates announced this program, there were 371 applications. I was on the review committee for this, and When I first heard there were 371 applications, I was concerned they were going to send them all to me. Luckily, didn't. We got that down to a smaller number, reviewed them carefully, and asked for 21 full proposals, of which 10 groups were funded. So there were 10 individual grants for up to $750,000 each, for up to 24 months, and we're just coming up to the end of that 24 months for that phase one now in the next few months. Phase two, it's expected that a smaller number of projects, a subset of those, 3 to 6 of them, would be funded going forward. There's $3 to $5 million of follow-on funding available, and hopefully that there would be some validated biomarkers or a validated biomarker coming out of that. You heard Robert Moritz's talk earlier. He's one of those grantees. Each of the grantees is assessing their biomarker or validating their biomarker against a series of reference materials that we're providing, really to ensure that there's not really high variability in the types of specimens that people are using, that in fact TB suspects are the control group and not healthy volunteers in New Jersey, and to enable analysis of data across projects. And so, for example, some of the groups are working on very similar questions, and it's helpful to have them use aliquots from the very same patients so they can compare their data. So Jennifer Gardner and I sat down at the start of this project and sort of tried to map out of the biomarkers that have been proposed or that you can think of, how do they graphically look if you chart them by sort of ease of translation into a point of care platform on the y-axis, and our confidence that it's actually a biomarker for disease, for TB itself. So just an example, the whole organism has a very high, I can't get this to work. The whole organism has a very high score on the confidence in biomarker. It's on the right-hand side. Obviously TB is TB. Whereas various metabolites, small molecules in serum, might be slightly easier to detect, but might have a lower confidence of actually being tuberculosis when detected. And of course, those plot across different matrices from sputum, urine, blood, and breath. So there are now funding efforts in each of these areas that have been circled. So I won't talk about these projects in detail, partly because they're confidential and partly because there won't be time here. But the investigators' names will be on there, and you can run into them or talk to them and get your own information for them to the degree they're willing to give to you. As Dr. Moritz already showed you, at the Institute of Systems Biology, they're doing some sophisticated mass spec analyses of reference materials to try to map out what are the TB proteins that can be detected in various matrices and at what concentration. Karen Dovus is, if you're familiar with exosomes, these are vesicular bodies that are generated by cells, infected or not. They circulate in high concentration in the serum, up to 10 to the 11 exosomes per mil. And they contain concentrated fragments of both host materials and, if there's an infected cell, cellular pathogen materials inside them, allowing you a concentration mechanism, if you will, for TB proteins ideally. So she's looking with mass spec, a similar type of mass spec as Dr. Moritz is using, to look for TB proteins inside those exosomes. The Feldheim group is trying to get a little more greater understanding of LAM and its availability in serum and in urine, and especially what kind of LAM fragments there are, how they might best be detected, whether they would be complexed with proteins or other molecules that would make them difficult to find. The Ochsner group at SomaLogic is using somomers. These are slow off-take modified aptamers. They're DNA constructs that you can use as ligands. And they've developed these to detect, they've developed somomers against 2,000 human proteins and put them on a chip so that you can screen serum, for example, for the concentration of 2,000 host proteins at one time. So they're looking at those and comparing TB patients and TB suspects to see if there is a signature of host markers that would be sufficiently diagnostic to either use by itself or to become complexed with, for example, serologic testing to help you understand or analyze those results. David Alon's lab is using magnetic nanoparticles that are functionalized with antibodies to try to pull TB out of sputum. David Anderson at the Burnett Institute in Australia is looking at novel antibody classes, including dimeric IgA, to try to improve the sensitivity of serologic testing. In Louisville, the Graham group is looking at volatile detection in breath. Obviously, these are complex clinical studies because we have to collect these in real time, ship them to Louisville. The Belial group is looking at some small molecules, metabolites found in serum that they've following off with some work from some of the TBTC studies where they identified a series of metabolites that decreased dramatically after starting with TB therapy, and following up on whether these are diagnostic, and also looking at some glycolic acids that have been identified both in sputum and serum. Antonio Campos Neto, his group in Boston, is looking at trying to validate 5 protein markers that they found in urine and have published on by using the Quanterix single molecule detection system there in Boston. And lastly, Todd Lowry has done a Herculean effort to synthesize a large number of lipids, lipoglycans that would be the predicted fragments of a number of lipids from TB, so that we can see if actually fragments of those might be more detectable than the whole molecules. Is developing, it is screening also for antibody responses to all of those synthetic molecules. So as you can see, some of these are pathogen specific molecules, and some of these are host specific, and some we don't actually know if they're pathogen or host. They're in different matrices: blood, urine, sputum, and breath, and some of them are proteins, lipids, proteins and sugar, or both organic compounds. So in summary, the Gates Foundation is currently funding 10 projects. They are coming to a conclusion now. They will be moving into phase 2 in the first quarter of next year, and try to validate and optimize at least one, if not several, biomarkers. Thank you.